Hello, everyone. Thank you all for joining. Welcome to the USACM Novel Method Fall 2025 seminar. I'm Paola. I'm currently an associate professor of mechanical engineering at the University of Utah, and I'm the co-chair of the technical trust area. Along with Professor John Foster from UT Austin, we've been doing this seminar series for the past couple of years. The goal is to bring the community together and create a community of learners by just sharing our knowledge and openly discussing different advancement in the numerical methods. Typically, we feature two outstanding researchers from two different continents. Today we are fortunate to have Professor Paola Antonietti and Professor Arzani with us. Each speaker will typically speak about 30 to 35 minutes, followed by 5 to 10 minutes Q&A. And during the talk, feel free to write down your questions in the chat, or at the end, during the Q&A session, you can just simply unmute yourself and ask your questions directly. So with that, let me start introducing the first speaker. Our first speaker is Professor Paola Antonietti. She is the head of the Laboratory of Modeling and Scientific Computing, MOX, and a full professor of numerical analysis at Politecnico di Milano. Her research centers on advanced numerical method and computational learning techniques for the approximate solution of partial differential equations with applications across various fields, including computational neuroscience, engineering seismology, and subsurface flow simulations. She has authored 2 books and written over 100 publications in various international journals. She received the 2016 CIMAIS prize from the Italian Society of Applied and Industrial Mathematics and the 2020 Lions Award from ECOMAS. She is the recipient also of a 2023 ERC, which is funded by the European Union. With that, please join me and welcome our first speaker, Professor Antonietti.
So thank you. Thank you very much, Pania, for the invitation and for the opportunity to join this very, very nice initiative. What I would like to discuss today is some results in the framework of try to better understand the human brain, and in particular neurodegenerative disorder, through the use of advanced computational models and machine learning techniques. So first, before entering into the details, let me thank all the younger colleagues and students that are part of this project. So nothing of what I'm going to show today would have been possible without the joint effort of the younger researchers that are part of the project. And let me also share that whenever you are interested in trying to explore such a topic, we made available some basic software or open source software to better disseminate the results which are part of this project. So the idea is try to better understand, and ultimately the goal is to support clinical doctors in a better understanding the underlying mechanism, and ultimately better treat patients that undergoes some form of a neurodegenerative disorder. And from the medical viewpoint, neurodegenerative disease is an umbrella term that covers a wide range of pathological processes with very different clinical symptoms, which ultimately lead to cognitive decline and motor dysfunctions. It's quite it's well known within the medical community that certain class of neurodegenerative disorder are technically classified as proteinopathies, which means that the underlying process at the biological level is characterized by protein misfolding. And so at a certain time of a patient life, proteins, and I'll make this statement more clear in a second, start to misfold, to aggregate, and to spread through neural pathways with the so-called prion-like paradigm. And despite the fact that so the involved protein is a different and characterize a different neurodegenerative disorder, the underlying mechanism is the same for many proteinopathies. For example, in Parkinson disease, the involved protein is the so-called alpha-synuclein. Patients which are diagnosed with Alzheimer's disease, so this disease is characterized by two interconnected misfolding proteins. First, the beta amyloid proteins starts to misfold, aggregate, and then diffuse through neuron pathways. Then tau protein is triggered by alpha beta synuclein, and there's a second misfolding process. And there are many others. So Alzheimer's and Parkinson's are the two diseases with the most incidence, but there are many other diseases which are characterized by this prion-like mechanism. So the idea is to try to better understand, so try to use the universal language of mathematics and computational modeling to set up a framework that is able to reproduce such a process to better, to support medical doctors and ultimately, of course, patients. And why is so important? Well, so mathematics and computational modeling is more and more used in the framework of computational neurosciences. The reason is that neurodegenerative disorders are very long time frame diseases. So in the picture you see in the slides, which is just a schematic pictures, just to give you the idea, you can see, for example, the red line. So the first one gives you a qualitative description of how the accumulation of the amyloid beta, which is one of the proteins primarily involved in Alzheimer's disease, evolve over time. So on the x-axis, you see the progress of patient status from cognitively normal. MCI stays for mild cognitive impairment, which means that the patients or its relatives start to realize that there are the first symptoms and the last. So on the bottom right, you see the fully conclamated stage of dementia. On the y-axis, there's this shift from a normal concentration of a biomarker that measure the concentration of beta amyloid to an abnormal concentration. So this gives you a qualitative overview of how concentration of misfolded beta amyloid evolves over time. The clinical function is the orange line. So you see when you first start to observe the first symptom of the pathology, unfortunately, the concentration of misfolded beta amyloid is already at a plateau. And this is, at least by now, an irreversible process. Let's try to see whether technology can support an early detection. So, for example, you can have a look at the green line. This is a brain structure. So whenever an imaging examination can detect the deposit of beta synuclein, assuming that, of beta amyloid, assuming that we can, of course, undertake very invasive imaging exams, which of course is not the case. But also, even with, if it were possible to undergo advanced imaging, whenever the deposits are large enough to be detected, the concentration of beta amyloid is already very high. So the point is that this misfolding process and the evolution through time take place, so starts much before the symptoms appears. And much before, so with the known level of technology we do have today, we are able to detect deposits of misfolded beta amyloid. Also, for that's the example I use is in the context of Alzheimer's disease. For example, for Parkinson disease, it's even so the scenario is even more dramatic, because nowadays there are no tracers which are approved for clinical practice that can be used through a PET imaging to measure the deposits of alpha-synuclein. So this, said in other words, this means that almost all the diagnoses of Parkinson are made based on indirect measure. So there's no way to trace the deposit of alpha-synuclein in the parenchyma. What I didn't tell you is the time scale of this phenomenon. So this phenomenon can last from 20 to 30 ages, which means that a patient that today exhibits the first symptoms of mild cognitive impairment starts the misfolding process up to 30 years ago. So that's why medical doctors are particularly interested in trying to have predictive physics-based models, maybe augmented with data and computational learning techniques, to better understand the mechanism that drives and sustains neurodegeneration. Which is not only connected, or to be more precise, which is strongly interconnected with many other processes in the brain, which is a very complicated structure, both from the viewpoint of the mechanical characterization, but also because of the interplay of many different phenomena. So due to neuronal loss, it is well known that patient affected by neurodegenerative disorder goes through a loss of brain material. So typically, these are post-mortem images. So what you can see is that so in the very late stage of the disease, there is an important shrinkage of the gray and white matter, and this results in very enlarged ventricles. So this black, I'm not sure whether you can see my pointer, but these are ventricles in a healthy patient and in a patient with an advanced stage Alzheimer's disease. And why this is important? Because we do have, all of us, a so-called waste clearance system. So through cerebrospinal fluid, which is in an healthy patient is completely rechanged 3 times a day. This is used as to wash out all the toxic substances which are produced naturally and physiologically by brain functioning, including toxic proteins, which are misfolded. And it's also known that due to aging, the performance of our waste clearance system deteriorates. It's also known that in patients with some sort of dementia, such a system is more impaired. And this is somehow a sustained mechanism, because if the clearance system doesn't work as expected, this of course sustain the deposit of toxic proteins. So an imbalance in the production and wash out of the cerebrospinal fluid serves and is a triggering mechanism of misfolded protein aggregation. And that's not at the end of the story, because there's also vascular dysfunction. So there are many biological and physical processes that are self-reinforcing mechanism where the exacerbation of neurodegenerative disorder is sustained and triggered by other factors. From the mathematical and modeling and computational viewpoints, what are the challenges? So as I said, there are very different time scales in which these processes takes place. So prion distribution, so the mechanism of misfolding, aggregation, and diffusions happens on decades time scales, and also remodeling due to neuronal loss. Then it is also known that so the waste clearance mechanism, and in particular the washout of toxic substances, happens at hours timescales, and in particular so during sleep hours is the time frame of the day where most of the where we are more prone to wash out toxic substances. And then there is, of course, the heartbeat, so second time scales, because this is where oxygen is provided to the brain. And there's a last, even shorter time scale, which is, so in some of neurodegenerative disorders like Parkinson and Alzheimer, so the neurodegenerative disorder triggers the formation of an impaired propagation of the electric signal, so seizures. In some patients, this is the first symptoms of that something is not working properly. So you see, to have a comprehensive picture of what are all the mechanism and interplay that sustains neurodegenerations, we should be able to cover a broad range of time scales. We are not yet there, so we are trying to attack the problems at the different time scale with the ultimate objective to be able to better understand the interplay between all these mechanisms. So what I would like to discuss today, I think that I'll mostly focus on how to build up a generalized model that can describe through a fully physics-based approach the protein spreading. And then if I have time, I'll say a couple of words on brain perfusion and waste clearance mechanism and on epileptic seizures. So the simplest possible model, this is one of the simplest example of a conversional system. So to describe the process of aggregation and diffusion of toxic proteins like prions in our case, then it has to be specialized according to the neurodegenerative diseases that we are interested in is the so-called Fischer-Kolmogorov. So here C, which is a function of space and time, is the relative proteins concentration over the total number of proteins. So you have to assume to have a little piece, unitary length of brain parenchyma, looking at the picture very far, and you want in like an homogenized problem, have a look at from very far to see the evolution of the relative concentration of misfolded proteins. There are two ingredients which are very important here. So first, the diffusion tensor. In this kind of applications, it has a very established expression. So you have a part which is isotropic, but most of diffusions happens along axonal directions. And how diffusion takes place in the brain can be estimated through diffusion-weighted MRI. So this data show how the diffusion tensor looks like is something that can be measured in a patient-specific manner. And then there is this coefficient here. This is alpha. This is known to be the conversional factor. So it's the ability of a misfolded protein to, sorry, let's say in this other way. It's the velocity or the ratio at which healthy proteins are converted into misfolded ones. This is a parameter that cannot be measured because it encoding all a number of considerations that happens at the very small scale. And this is a organ scale model, which is the characteristic space length we are interested with. And now, of course, there's no way I can solve this in a patient-specific brain geometry, so I need computational methods. And what we are particularly interested in are computational methods that to some extent extend the paradigm of classical finite element methods on tetrahedral and hexahedral grids to arbitrary shape polyhedral meshes. Why? Well, because this is an example of a geometry which features an extreme level of geometric complexity with very heterogeneous materials that has to be properly described in order to capture the underlying physics. And the idea is that if you have these very small features, let's say features which are like the cerebral sulci, which have characteristic length scale to be epsilon, in order to be able to describe all these features, you need the mesh with the characteristic size epsilon, which makes the underlying computational effort unfeasible, especially if you want to make hundreds of simulations in patient-specific setting. So polytopic grid offer the required level of geometric flexibility in order to save the number of levels required to correctly describe all such complicated features. And we have recently developed an algorithm based on graph neural network in order to be able to automatically construct such a meshes respecting the underlying heterogeneity of the materials. So let me switch. Now, I won't enter into the details of computational methods, but the idea is that you can write using a so-called discontinuous paradigm. So my solution is going to be a piecewise polynomial in each of my cells without any continuity constraints, so that I'm free to adjust the mesh size and the polynomial degree according to the level of accuracy I want to have in the different brain regions. And so I'll skip the details, but the idea is that after the discretization in space and then in time, I have a huge system of ordinary differential equations if I treat the nonlinearity in a proper way. Let me also make a comment. So if so physically speaking, my solution of my continuous problem is a relative concentration. So it is it should be something between 0 and 1 because it's a relative concentration. You can prove that this very important property is satisfied at the continuous level, but this is not for free at the discrete level. So when you move from your system at the continuous level to the discrete counterpart, you might lose, for example, positivity. So what we are working on now very intensively is to make sure to extend or to reformulate our problem at the discrete level in order to be able to be so-called structure preserving. So to be able that our scheme incorporates naturally the physical constraints that are that holds in general at the continuous level. But I won't enter into the details. Let me show you some practical realization of the model I propose. So that's a simulation of the evolution of the Parkinson disease. So here CH, so my computational solution is the concentration of alpha-synuclein. So we start from a seed, which is here at the very bottom of our brain. That's one of the typical regions where Parkinson is known to start to evolve. And this is the evolution of the concentration of alpha-synuclein through a time frame of 20 years. Now, how we can embed patient-specific data into our simulations. This is an example I want to discuss of a patient, an 83 years old male patient with a diagnosis of Alzheimer's disease, which undergone through the whole series of diagnostic steps. That's not always the case for all the patients that are diagnosed with neurodegenerative disorder. So from structural MRI, we can reconstruct the geometry that serves as one of the ingredients of our model. From diffusion-weighted MRI, we can reconstruct patient-specific axonal directions. These are very important to set up the diffusion tensor in our model. And from a PET, we can measure the initial concentration of beta amyloid for the patient at the stage of the disease. And now that's the result. So here, the initial time, this is not simulation, this is data reconstructed from PET, and that's the evolution of the relative concentration into three brain sections. The horizontal plane is the first row, coronal plane the middle row, and sagittal plane in the last row. And please notice that here, unfortunately, we have a much shorter time frame because at the moment of the diagnosis, the pathology was already at quite far stage. Now, that's not the simplest mathematical model you can set up I won't enter into the details, but the point I want to make is that so there's a family of more complicated models from the mathematical viewpoint, and to some extent more informative. This is an example. This is the heterodimer model, where you work with both healthy and misfolded protein concentrations, and we can by now handle also such a more informative type of models. And this is an example where you simulate at the same time both native proteins and misfolded proteins, again for a patient with Parkinson disease. And from a qualitative comparison between the space and time evolution of the misfolded aggregates, these match, at least qualitatively, the typical evolution of Parkinson, which starts from the lower brain stem and then moves in the mesocortex, and in the final stage of the disease arrive in the neocortex. This is a result which tried to match, also to validate the model against clinical data. So the blue line is the result of the numerical simulations. Please notice that for Parkinson, so the first two stages are where no symptoms can be appreciated. The symptomatic stages these are called Brack stages. It's a way of classifying the level of severity of the pathology. This is the final stages of the disease, and the clinical data that can be reconstructed measuring biomarker abnormality are reported in the black crosses. And of course, there's not a perfect match. This is a model with a certain number of simplifications, but at least the evolution of the symptoms is clearly captured, or the severity of the disease is clearly captured by the numerical model. And I think I'll conclude just saying that this is not the end of the story. Well, actually, it's only the beginning. What we are working now is try to couple a model of diffusion of the proteins to what I mentioned at the beginning. So also taking into account mass loss, which is very important because this is a strong effect in the balance of fluid. So cerebrospinal fluid flows and waste clearance mechanism. That's a simple model we started from, where now we have both proteins concentration, the tissue deformation displacement, and how mass reduces due to the accumulation of misfolded proteins. Let me just show the final results. So this is a tau spreading in a patient affected by Alzheimer's disease. Let me show you once more. That's the volume loss variable, which is known to start approximately after 10 years of the initial evolution of Alzheimer. And this is the simulation for the corresponding tissue deformation. And with this, so that's a snapshot after 15 years. So the evolution of tau protein spreading, the corresponding volume loss variable, and tissue deformation. And I think my time is over, right? So let me go to the conclusion. We are working on trying to set up a comprehensive mathematical models to better understand the mechanism that sustains and triggers neurodegeneration. There are, well, at least from my viewpoint, very fascinating questions, both from the modeling viewpoint, challenges at the numerical levels, and then at the more applicative level. What I didn't discuss is, so there's a lot of source of information in the data. we are now, for example, in cognitive test, which is the baseline for exams for patients that are diagnosed with a neurodegenerative disorder. Now we have ODE model that integrates physics-based models to better try to better understand the evolution of the pathology. And we are also working in integrating how neurodegenerative disorder triggers epileptic seizure, which I didn't have time to discuss. But I think I'll stop here because my time is over, and I thank you for your attention.
Thank you so much for the wonderful and fascinating talk. Thank you so much. It was really amazing. Thank you. I guess now it's time for question. Whoever has a question, you can just unmute yourself and ask your question directly.
Yeah, how do you include the time dependence of the parameters in your model? Presumably, these parameters are affected by things like the chemical environment in the brain, which is going to change as people age.
So thank you. Thank you. This is an excellent question. So what we are doing right now, so in the literature, most of the models assume that this constant, which cannot be estimated. So what we do usually is to do inverse parameter estimations only for those patients for which we have more than 2 sets of images. It is assumed constant, but that's not, well, at least in our experience, this is not definitely the case. So alpha should vary both as a function of space, so in the different brain region, but also as a function of time. So what we are trying to do right now is move to reduce models on graph that allows us to better estimate how alpha varies as a function of time and space, and then to use such an estimation in fully 3D physics-based models. But that's definitely an open question.
Okay, thank you.
Thank you.
Anybody else? I have a question. So so it seems that most of the focus has been on this, especially I guess on this slide. So you are looking at this diffusion tensor, but it's how do you account for the change in the structures? Like, because you mentioned that throughout the years there are changes. There are I know that at the last slide you said that you are trying to I guess couple them with other physics, but in my mind, when I guess there is a chemical reactions taking place, and then there is a change in those empty spaces. I like to call them, I guess, pores. There, how do you account for this diffusion tensor? So it should be impact on the diffusion, right? Because it's an important parameter.
Yes, thanks, Pania, for the question. Definitely. So this can be. Let me go to the other slide if I can. So the point is again the lack of data, in the sense that if we do have diffusion weighted MRI, for example, at different stages of the disease, we can fully reconstruct how the axonal directions changes and then incorporated this due to structural changes. But unfortunately, well, or fortunately, it depends, only few patients undergoes a full set of exams. And only a few, a small set of these and few patients do this in a longitudinal fashion. So only for few patients we do have this full set. So to set up the diffusion tensor, we basically need diffusion weighted MRI. Only few patients do this set of this panel of images test throughout the evolution of the pathology, because these are invasive tests. And if the patient is cognitively impaired in a substantial way, basically the medical practice is mostly based on cognitive tests. This is why I was mentioning at the end that we are working hard to incorporate all the knowledge which is in a cognitive test, like that are part of the clinical practice, because imaging is a very invasive test. And for example, so only few patients undergo to the PET scan, which is a way to measure the deposits at the initial step of the pathology, or at the initial step of the diagnosis process. If we don't have the data, either we infer it starting from the data we have, which of course is a rough approximation, or otherwise we have to move to reduced type of models, for example, on graph, where you have less impact of how diffusion tensor, so the role of the diffusion tensor.
So don't I guess based on your experience working in this field for long, don't you think that the lack of the I guess resolutions of the experimental technique is one of the reasons that we are diagnosing these at very late stage? Do you think that if we look at it at the smaller scales at the cell level, or if we have those techniques that we can, I don't know, MRI with a much higher resolution, would that be a possibility to?
That's a difficult question, in the sense that, well, at least what neurologists told us through our collaborations, I mean, even if we would have the technology, the real point is that when the symptoms appears, it seems that the process, so the chemical process, is already too far away.
Oh.
Now, at least in the Italian system, to be honest, I don't know how it is in the US system. I think because so a huge change is going to hopefully happen in a few months or years, which is blood biomarkers. So now, at least in Europe, in the Italian system, blood biomarkers, so that you measure through blood some sort of markers which are known to be correlated to the deposits of beta amyloid, at least for Alzheimer's. These are not approved in the clinical practice. So they can be used for research purposes, but not in the clinical practice. I'm not sure in the US they are already approved, most probably yes. This is going to be perceived from the medical community as a major breakthrough, because of course the screening with the blood test, you can screen a wider set of population, because of course you cannot ask an healthy person to undergo a PAT. This is wouldn't be sustainable for the system, right? Because only few hospitals has the equipment to do that, and it's always a matter of risk and benefits. But with blood biomarkers, it might be that since it's much more affordable and easy to be done, also in terms of the impacts on patients, this might be really a turn of paradigm for early diagnosis.
Thank you. So one of our listeners is asking, I get Keratin, saying, thanks for the great works, Paula. Have you worked on any optimal treatment models connected to the diffusion models you developed?
Thanks, Kiratan. I'm not sure I understood the question. Optimal treatment means supplementing the model with, like, for example, external sources that try to model the effects of drugs or possible treatment. If I understood it correctly, the answer is not yet.
Thank you so much.
That would be definitely very, very interesting.
Yeah. So if anybody else has question, you can, I imagine you can directly reach out
Sure.
to her.
Yeah,
So
sure.
I have many more. I'll be one of them. So, but for the sake of time, I guess we need to move on to the second speaker. So thank you again, Paula, for the great talk.
My pleasure. Thank you.
Thank you. So the second speaker is Dr. Amirhossein Arzani, and he's currently an associate professor at the University of Utah, where he has a joint position at Scientific Computing and Imaging Institute and Mechanical Engineering Department. He received his PhD in mechanical engineering from UC Berkeley, and his research involves developing various computational mechanics techniques and tools, as well as data-driven techniques for different applications, including biomedical flow and biomechanics. He's the recipient of the NSF Career Award, as well as NIH Trailblazer Award, and also one of the most prestigious early career awards known as Presidential Early Career Award for Scientists and Engineers, in short, PCASE Award, which was given to him by President Biden. So with that, welcome, Amir, and we are ready to hear your talk. Thank you.
Thank you so much, Panya. Let me share my screen again. So today I'll be talking about interpretable data-driven modeling. So as we all know, deep learning has become really important in scientific computing and physics-based modeling, and it's found a wide range of applications. But there are two key challenges when you want to apply deep learning to scientific problems. The first one has to do with interpretability. And interpretability is important because you want to, at the end of the day, understand the systems that we're studying, and it's also useful in debugging our models, and at the end of the day, provides trust in the models that we want to deploy. And the second challenge has to do with a generalization to out-of-distribution data, or really extrapolation. And I would argue that that's also desirable at some level, especially when we want to deploy our models. Sometimes in some applications, we don't exactly know the boundaries of our training landscape. So it's desirable to have some level of robustness to some level of extrapolation. So, and there's also evidence in the literature that the more interpretable models typically tend to generalize better. So in this specific talk, I'm going to be focusing on interpretability. So that brings us to the field of XAI, explainable AI. We can divide it into two classes of problems. We have the by design problems, where we integrate interpretability from the beginning into the architecture of our machine learning model, so by construct. And then we have the post hoc approach, where we take an opaque neural network, we probe it, generate data from it, and in a post-processing step, we interpret the opaque neural network. So in this talk, I'm going to be focusing on by design interpretable AI. And my goal today is going to be discovering nonlinear relationships, either analytically from the data or by designing new architectures that are interpretable. And the vision is going to be using building blocks of neural networks. So essentially what's made neural networks successful, so things like global optimization, additive, we want to use this technology with minimal reliance on those opaque neural networks themselves, so in order to get the interpretability that we desire. So that's going to be the vision. Okay, So this is an outline for my talk. I'm going to show you three applications. The first one is going to be dynamical system identification. The second one, interpretable nonlinear reduced order modeling. And finally, interpretable operator learning. And this three parts of the talk, the three stories I'm going to tell you today are going to be motivated by three key equations in scientific machine learning. So I would argue that these three equations are very important equations in machine learning. So one is sparse regression, the other one is PCA, and finally is a simple functional linear model. So these equations are really fundamental and important in scientific machine learning. But what we're going to be doing today is that we're going to be looking at these equations and asking ourselves, can we do better using the modern computing technology we have, such as PyTorch? Can we do better than these equations? And can we do better in an interpretable manner? So that's going to be the three stories that I'll be telling you today. So let's start with sparse regression. The problem here is that we have some data, typically trajectories of our system, and the goal is to use that data to build an analytical equation, typically systems of differential equations that match our data. So we can use deep learning to approximate our data, but that's not an interpretable approach. So on the interpretable side, we can use either symbolic regression, or we can use the popular SIND approach, sparse identification of nonlinear dynamics, which I'll talk about. And a motivating problem, which is a very new research direction in my group, is actually cancer systems pharmacology and digital twin modeling. And the idea here is that, from patients that have cancer, we can get their samples from their tumor, and we can do in vitro testing to obtain subcellular protein concentration data, as well as cell scale data using methods like cell sequencing and proteomics. And the question you want to ask is that, can we develop computational models that can take this type of data and fit governing equations to them? Because once we have mechanistic governing equations, then we can use them to optimized treatment, which is an open problem in cancer. So specifically, I'm going to be focusing on subcellular signaling modeling. So that's going to be finding a systems of nonlinear ODE represented by a network. And then that's going to be coupled to a cell scale model, which represents the interaction between different cell phenotypes. Okay. So, but before we get there, so the question is going to be, can we build these mechanistic models from input data? So now the approach we're going to be using is going to be SIND. And if we look at the alternative approach, symbolic regression, so symbolic regression takes data and essentially it searches for all mathematical expressions and their composition to fit the data. So this is really, it's an expensive problem. It uses typically genetic algorithms to do the global optimization, and sometimes leads to complicated expressions. On the other hand, we have the SIND approach, which is a much more structured approach to finding governing equations from data. And the way it works is that you provide a library of candidate terms, so the analytical terms that you think could be important, and then it formulates a convex optimization through a sparse regression and finds the governing equation from data. Now, the problem with SIND is that if you have nonlinear parameters like Fourier frequencies, growth decay rates, like exponential bandwidths, you need to know these nonlinear parameters beforehand. And that's challenging to guess or know. So in ADAM-SIND, the way we approach it is that we shift the convex optimization problem to a global optimization problem with ADAM optimization. And we also learn those nonlinear parameters. So we don't need to guess the nonlinear parameters. And also we do a fully adaptive sparsity promoting strategy, which, as I'll explain later, gives us machine precision accuracy. Okay, so this is a review of the SIND framework. So the idea is that you have a system of governing equations. You have the solution, so you can measure the solution. So you know x as a function of time, so you already have your answer. And then your goal is to find a governing equation, to find f of x. You build a library theta of candidate terms. So any candidate term you think are possibly important, you throw it at the library. And the challenge is that if you have nonlinear parameters like powers, frequencies, and exponential bandwidths. If you have these types of parameters, you need to guess them in your library or include a range of values in your library. And then you solve this sparse regression problem to select a sparse set of these candidate terms, linearly add them together, find their coefficients, and represent that as your model. So typically, the sparsity strategy in SIND is an identity matrix for gamma. So you uniformly penalize for sparsity on all your candidate terms. So now, our approach, Adam-Cindy, there are two key differences in Adam-Cindy. The first one is that we also make the nonlinear parameters trainable. So we don't guess the nonlinear parameters, we solve for them through the Adam optimization. So that's the first big difference. The second big difference is that we don't do uniform sparsity. This gamma matrix that imposes sparsity, we define it in a candidate-wise manner. So every term can have its own penalization, and then we learn that as we go. So that's the two key differences. And to formulate the problem, we have two strategies. We can either do a min-max optimization or a joint minimization approach. And what we've learned after going back and forth between these two approaches is that the min-max optimization is more difficult to converge. It requires like tricks like weight decay, but the min-min approach is much more robust. And the min-min approach works because what happens is that the adaptive sparsity parameter, you might think that because you're minimizing everything, doesn't it just go to zero? But the interesting thing is that it goes to zero for correct terms, but it converges to a constant value for incorrect terms. And this is what we see in all the test cases that we investigate. And that is actually why we get machine precision accuracy in Adam-Cindy. So if you take a closer look, this is the optimization problem we're solving. And because it's a minimization problem, it drives the sparsity promoting parameter gamma towards zero. And what that happens is that the L1 sparsity promoting term goes away. You only left with your data fidelity term. So you do a perfect machine precision fit to your data. But then for the incorrect terms, what happens is that because the coefficient goes to zero and you do thresholding, you zero it out, then the gradient becomes zero, so it doesn't change anymore. It just saturates to a non-zero value. So that's the trick why Adam-Cindy works so well. Okay, so the strategy is that we're penalizing only the incorrect terms while we spare the correct ones. And that helps us get very accurate results. So we have looked at multiple problems. I'm just, in the interest of time, going to focus on a couple. So this is the so-called ABC flow problem. So this is the ground truth equation. It's an incompressible flow. So you can think of this as XYZ velocity. And it's a benchmark problem in chaotic advection. It's an incompressible flow. And we actually took this benchmark problem and we made it even more complex by making the frequencies different for every term. So now, if you look at the classical Cindy method, so this is Cindy with knowing the exact frequencies. If you happen to know exactly the nonlinear parameters in your system, the classical Cindy method works perfectly fine. No problem with that. But if you don't know the exact frequencies, which is the case in 99% of the problems, and you guess them, and your guess is not right, which again is very likely, you get a dense system like this, which doesn't match the ground truth. But in Adam-Cindy, we can optimize for it. So we can solve for it. So we find not just the coefficients, but also the frequencies. And you can see I'm placing pi here, not the digits, because we actually find this frequency up to machine precision accuracy because of what I just explained. Okay, we can also do this for PDE problems. This is a wildfire transport problem. This is a nonlinear advection diffusion reaction problem. So you have a reaction term that's nonlinear, that has a nonlinear parameter. So in Paola's talk, you also had a reaction term. We had a reaction term there too, but there it was a linear reaction. But in some applications, we have nonlinear reactions that makes it more challenging to infer the parameters. But again, we can see that Adam-Cindy can find these nonlinear reaction as well. Okay. So now back to our cancer motivation problem. So this is the ground truth equation, how it looks for cancer. It's a large subcellular systems of ODE. These are all nonlinear ODEs that you can summarize in this diagram here, what they represent, reaction between different proteins. You have these Michaelis-Menten terms, which gives it nonlinear, and that's coupled to the cell scale model. So here we have resistant cells that are resistant to drug, sensitive cells, and differentiated cells. And the output of this cell scale model tells us how much cancer grows, okay? So now we take data, in this case, it's a clean synthetic experimental data. We do different sorts of perturbations to generate data. And then we want to take the trajectories of this data. And our goal is to find these equations that you see on the right-hand side. Okay, so this is much more complicated than the benchmark problems I just presented. Okay, now here's the challenge. In systems biology, a lot of these trajectories of these different states are correlated. So that makes system identification very, very difficult in biology, okay? So now we were not able to do a one-shot identification like the benchmark problems I presented. So what we did was a two-stage approach. We took the data, we applied Adam-Cindy to it, and that gave us a clue to eliminate a bunch of candidate terms. Then we took the remaining terms, and what we did from there was that we basically looked into all different combinations of the remaining terms and just did a simple nonlinear regression there, and used the AIC score to select the best model. Okay, and what we saw was that Adam-Cindy could reduce the total number of models that we had to test by 90%. Okay, so this is some representative results. So these are some of the states. These are some true equations, and these are the equations that we find. For the 18 proteins, in 100% of the time, we were able to find the correct governing equation. But since in this second stage we used nonlinear least squares, we had some error in the parameters. So we had up to 1% error in the parameters that we found. Okay, so now let's go to the second part of my talk, which is about nonlinear reduced order modeling. So in this part, I'm going to be focusing on this very, very simple equation, PCA, Machine Learning 101. If you take any machine learning class, this is where it all starts, this simple, beautiful PCA equation. Okay, and the motivation for PCA in scientific machine learning really all started with reduced order modeling, and I would argue in fluid flow, probably in the 80s. And in fluid mechanics, we call it POD. And we take a spatial temporal data, U as a function of space and time, and POD gives us this beautiful decomposition in space and time. Linear, beautiful, as simple as we can get to find a reduced order model. Well, but you can argue that in complex problems, you need many modes, right? So then you have the autoencoders, neural networks, where you do this in a latent space. It gives you more accuracy, fewer modes, but you have challenges in interpretability. And also you can't get both latent space in both time and space. Okay, so we talk about those challenges here in this paper. And there's some effort, of course, to fit a dynamical system in the latent space by some groups. But the method that we propose is another strategy. We call it DESMO, decomposed sparse model optimization. And the idea is that I want to get POD-like interpretability. I want to get equations that look like this, as beautiful as this, but with better accuracy. So that's my goal. Okay, I don't want to use neural networks. No neural network here. So can I do that? Can I extend POD to nonlinear equations by keeping the analytical form? So again, this is motivated by what I talked. We have Cindy, we have Adam-Cindy, which, Adam-Cindy, the motivation was to do global optimization, find the nonlinear parameters. We also have this other global optimization strategy, which I really like, optimizing a discrete loss, ODIL, where it's an alternative to PIN, where you have discretized 5 different equations, and you optimize all the stencils in a global optimization setting using Adam optimization. So now, DESMO sits somewhere in between. It's kind of motivated by all these techniques. And here's the idea. In PCA, I have my spatial temporal system. I decompose it into some spatial modes and temporal equations. I get an equation that's as beautiful as this for 4 modes. Now, what if I want to be more accurate? Now, this is where DESMO comes. So what I do in DESMO is that I take, like, for example, 2 base POD modes. So here I have phi1 and phi2. And then I say, okay, I'm going to stop here, and I'm going to build the nonlinear extensions of phi1 and phi2. This is all the nonlinear extensions that you're seeing here. And what I'm going to do is that I'm going to do sparse regression. Okay, so I'm going to solve this sparse regression problem. I'm going to pick some of these nonlinear terms. Okay, I'm going to learn which ones to pick to, at the end of the day, get an analytical equation that looks like this in space and time that's building off of my base POD modes. And those base POD modes could also be evolved during the optimization. And I'm finding some nonlinear extensions. Okay, so that's my goal. I want to replace this with this one. Okay, so now here's an example of a flow over a cylinder. So in this example, I took 2 base POD modes, phi1 and phi2, so these 2 guys, and I built and I searched using sparse optimization with Adam. I searched for nonlinear extensions of them. Okay, and ultimately what I get is something like this. This is the final equation that I get. So you can think of this as an analytical equation representing your spatial temporal system. And what I find is that I only had the base POD mode 1 or 2 that I initialized, but I find with my nonlinear method, I find the higher order POD modes as a function of the base POD modes, slightly per term. Okay, so I'm able to discover the higher order POD modes without even using them, using this technique. And when I compare the errors, I can see that with DESMO and 2 base modes, I can get the same error as POD with 4 modes, which should make sense based on what I just said. Okay, we can also do this in more complex problems. This is blood flow in a brain aneurysm. So I do a lot of blood flow problems in my group. This is a chaotic pulsatile spatial temporal system, and we can get an equation that looks like this. So this is with 4 base modes. I can get an equation that represents my spatial temporal data. So in this example, what we see is that we find some new modes that do not necessarily look like higher order POD modes. Okay, now the problems I have here are periodic in time. So what I can also do, I can enforce the temporal coefficients to follow a Fourier series. So if I do that, I'm gonna in the previous approaches for the temporal coefficients, I'm optimizing them. I'm finding them in every time step. If I want, I can fit an equation to them as well. That's easy. But another approach would be to use a Fourier series for them. So because the problems I presented to you are periodic in time. If I do that, it's going to give me less parameters to optimize. I'm going to now only optimize for frequencies and the coefficients. And by construct, I'm going to be periodic in time. So once I compare all the methods, I see that Desmo Fourier gives me less error, but in terms of interpretability, it generates more terms compared to the baseline Desmo method. So finally, if you want to couple this to a neural network, it's possible. All of this is in PyTorch. It's compatible with neural networks. So we also tried, instead of initializing the spatial modes with POD, find them with an autoencoder. And this is a special type of autoencoder that does it in space. And we train everything, Desmo and the autoencoder, end to end. So it's all trained together. Okay, so that's also another way to go about it. Okay, so the final part of my talk is going to be about interpretable operator learning. And here, I'm going to be looking at this integral operator equation. Okay, so I'm going to be motivated by this equation, and can I do better from this equation? So in operator learning, the problem is that we're doing mapping between data, but in function spaces. So a typical neural network, when you want to map f of x to u of x, you discretize them. You essentially lose the information in x. So x is gone. You just discretize f and u, and you let the neural network map it. But the idea in operator learning is that you want to keep the information in x. You want to keep the function form when you do the mapping between your data. And the simplest way you can think about it is this linear integral equation. Zeta here is a dummy variable that you integrate over. X is your free variable. Your goal is to learn this kernel K. Okay, you want to learn a kernel K, and you map it to U. And X, you can sample, once it's trained, you can sample any X you want. So you can do undersampling, you can do super resolution. So X, you could sample it. It's your free variable after you train and learn this K function. So there have been multiple operator learning architecture proposed. Many of them are motivated by this equation. For example, the popular FNO method takes this equation to the Fourier space and does it. DeepONet, if you look carefully into DeepONet, that one is also, there's a link between this equation. There are also other methods, NKN, KNO, and we also had a generalized functional linear model. And what we did in our approach was that we took a library of integral equations and then through sparse regression added. So kind of like Cindy, combining Cindy with this equation to find the kernel K. Now, what about interpretability? So okay, if you want to, so the theme of my talk is interpretation. So how do I do that here? 2 approaches. The first approach is that these are integral equations. So if you do it with integral equation approach, like the GFLM approach that we have, you can use the additive nature of integral equations, and you can decompose the contributions of different regions of your input data F to the output data. So this is one way to go about it, which you presented this, my student Mojgan presented this work at USACM. But today I'm going to show you a second way, a second strategy toward this, and that's interpretable latent space modeling by doing coarsening of your data. Okay, so think of X as a free variable. Okay, you're free to choose X however you want and sample train accordingly. That's the idea. Okay, so this is the architecture that we come to. This is differentiable autoencoding neural operator, Arduino. And the idea is that I have a high resolution grid that I want to solve some PD problem. What I'm going to do is that I'm going to map it with an operator to a coarse grid. Okay, solve my problem on the coarse grid, and then go back to the high resolution grid. Okay, so that's the idea. You have a difficult problem. You want to learn how do you take the difficult problem, convert it to an easy problem, solve the easy problem, and then go back to your high resolution correct space. And you can do different applications of this. You can do nonlinear dimensionality reduction by defining the dimensional reduction as a coarsening operator, going from a high-resolution grid to a coarse grid. You can do geometric reduction, convert a 2D problem to a 1D problem, and you can do many to one functional mapping. So in the applications I'm going to show you, the encoder decoder are a combination of FNO and a convolutional downsampling upsample. Okay, so here's the first example. This is just static mapping. Okay, so I have flow-over cylinder, I have a high resolution grid, and I map it to a coarse grid. And I'm comparing it to other methods that can also be visualized in the latent space. So the key thing about our method is that this method, you can visualize the latent space. You don't get that in many methods, okay? You can visualize the latent space in space, okay? It's spatial visualization, not some modes that you don't have an easy interpretation of. So here you can actually plot the latent space and look at it because of the way we define this architecture. So this is exactly what I'm doing here. I'm looking at this latent space results, and what I see is that if I compare this to like another method, CNO, which also gives us interpretable latent space, because I'm defining this as a coarsening operator, my latent space is actually physically meaningful. I can see the rotating, the clockwise, counterclockwise rotating vortices for flow-over cylinder, whereas in CNO, you lose that type of information in your latent space. Now, the more interesting application of this is not just data compression, but actually solving a problem in the latent space. So here I'm placing a differentiable PDE solver on the latent space, and I'm training this whole thing together, okay? Now, the idea is this differentiable PDE solver is going to be dealing with a coarser grid, so that's easier. Number two, I can place a lower fidelity version of my PD in the latent space. For example, if I have the full 2D momentum transport equation, I can place a linearized vortex transport equation here. I can place an inviscid diffusion equation here. I can represent different versions, even a 1D equation in the latent space. So I'm going to see how it's all will look, how will my latent space look if I place a low fidelity PDE in the latent space. And this is the results. What we see is that if I do a 2D inviscid vorticity transport equation, sorry, this should be vorticity transport equation, and a 1D version in the axial direction, I can preserve the coherent vertical structures, okay? But if I do like a Stokes type equation, which is just diffusion, I get something that looks like this in the latent space. So now, I argue that this is still interpretable. So this method is always interpretable. And definition of interpretable, I can look at it and visualize it. Is this physically meaningful? No. So if I put in a model that doesn't have the fidelity in the latent space, I get something that's physically irrelevant, but I can still visualize that. I can visualize that. I understand what my model is doing. But if I put a more higher fidelity model, I get structures in the latent space that preserve the main physics of my problem. And I can compare the accuracy of all these methods on test data. And at the end of the day, the latent space interpretability in this approach is governed by the PDE that I place in the latent space. Okay? So the final example I want to show here is that we can do also geometric reduction. So here's a 2D blood flow problem. I can take a 2D grid and I can convert the 2D problem to a 1D problem in the latent space by sampling X, okay? So here I'm learning how to go from a 2D transport problem to a spatially averaged reduced order 1D equation. So it's a geometrically reduced order model. And I have the 1D vorticity transport equation along the axial direction in the latent space. And I can actually visualize the latent space. This is how it looks like. This is how the latent space evolves in time. It's just a function of X. This is how the latent space looks as a function of X. So I can look at it, I can visualize it, and then I can see how my results look. So this is here a nice problem where we can learn how to do geometric reduction. And finally, we can also do this in 3D problems. This is the 3D coronary artery example. We can also do, here we're doing the pressure Poisson equation. I'm not going to get into details, but this is to show you that we can also do this in a more complex 3D problem setting as well, where we solve a 3D PDE in the latent space. Okay, so to summarize, I've showed you that IM-SINDI, an enhanced version of SINDI, can enable simultaneous optimization of nonlinear parameters, and it can give you machine precision accuracy, at least in the benchmark problems, and gives you a more flexible framework for finding interpretable governing equations in an analytical form from data. So DESMO, the second part, was an adaptive nonlinear extension of POD, which gives you better accuracy, but it keeps the analytical form. You can still write it on a paper, and it does it with less base modes. Okay, it just searches for extensions of those base modes. And then finally, Dyno was an interpretable latent space differentiable solver that I can learn how do I take a complex problem, a high fidelity PDE on a high resolution grid, how do I convert it to a coarse grid and place a lower fidelity PDE and solve it there. And I can still visualize it. So it's all interpretable. I can visualize what I'm doing in my latent space. Okay, and the big picture here is that modern global optimization, together with differentiable programming, has really high potential for interpretable modeling of nonlinear systems. And in all of these methods that I showed you, they were all implemented in PyTorch. You can couple all of these with neural networks, with different types of modern approaches. So there's a lot of opportunities here for this type of model. So with that, I'd like to thank funding, and also the 3 parts that I presented, you can find them online. The second paper will be online very soon. And yeah, thank you, everyone.
Thank you so much. Thank you, Amir, for the wonderful talk. It was really, really nice and informative. So now I guess I'd like to open it up for questions. Maybe I'll start with the questions that they are already on the chat. So Whiten is asking, what advantages does the Adam-Cindy framework offer in terms of interpretability and computational cost when we compare to neural ODEs and PINs?
Okay, so neural ODs and PIN. So in neural OD and PIN, in both of them, you need neural networks. Here, you don't need neural networks. Okay, so in neural ODs, in PINs, you're optimizing, I would say, thousands, tens of thousands, if not hundreds of thousands of parameters. Here, you're only optimizing the coefficients and the nonlinear parameters. So how many parameters? Maybe on the order of 10 parameters, or depending on the complexity, or maybe 100 at most. Okay, so that's the first thing. You're optimizing far less parameters, and you're not using any neural network. So pins, you can do system identification with pin, but you need some clever tricks. You can't directly just use the vanilla pin for system identification. And also for neural operators, which we also worked on neural ODEs, you get you have some level of interpretability, but you're going to have a neural network as one of the terms. Okay, so you're not going to have the full analytical form. So maybe you have some analytical forms in your equation, but then there's a neural network sitting in place of one of the terms. Maybe like your closure problem, right? So I would say for neural ODEs, it's semi-interpretable, right? Not fully interpretable. Yeah.
Thank you. So another question is coming from Kira, and he's asking, how sensitive is Adam-Cindy to noise? More specifically, the final symbolic form itself, are they sensitive to change in data?
So very good question. So there's nothing that we found in Adam-Cindy's architecture that provides any advantage compared to the basic Cindy approach related to noise. So if you want to use noise, I would say you could think about integrating Adam-Cindy with the weak Cindy form, which is shown to be more robust to noise. You can do the integral equation, weak Cindy, and then apply Adam-Cindy to it. And actually, we have a paper where we do something like that on integral equations in operators. That would be one way to go about it. So using the weak Cindy form and then applying Adam-Cindy to it. And the other way would be that you deal with the noise filtering in a pre-processing step. But the Adam-Cindy by itself doesn't give advantage. I think you have to couple it with weak Cindy to get some advantages, but that shouldn't be that difficult to do.
Okay. The other question in the chat is also coming from Whiten. Can the Desmo framework generalize to 3D flow through cylinder or unsteady turbulent regimes? I guess you touched on it a little bit, but maybe if you want.
So in Desmo, I showed you 2 examples. One of them was 3D spatial temporal system. So the blood flow example I showed you was a 3D blood flow problem, but we do have a turbulence example in the paper. So I didn't present to you. So we took the Johns Hopkins turbulence database. We actually did a slice. So it's a 2D turbulence. So turbulent channel flow. So we took the turbulent channel flow from Johns Hopkins database, and we also showed that you can use DSM on turbulence data. So the paper should be online within a few days. It's accepted. It will be online in less than a few days. So you can see in the paper once it's online, the turbulence example that we applied.
Thank you. So that's it from the chat. So now if anybody has a question, just feel free to unmute yourself and ask directly.
Can I go ahead and ask a question?
Yes, please, go ahead.
Hi, I'm Sushanta from Michigan Tech. Great talk, thank you. So I was wondering, I would like to interject on the point that you answered related to comparing against FNO and other type of physics informed model. So since what I understood from your work, that you are trying to solve the PD in lower dimension, or if you do POD, you would need some data to start with.
Right, of course, of course. There's no free, of course, all of these, none of these are purely, all of these you need data. So the idea is that for a certain, and I want to caution everybody, you cannot extrapolate. You cannot extrapolate. Okay, these are, the first talk I presented in Adam Cindy, if you hit a mechanistic model, there you're good. Okay, if you're hitting a mechanistic model. But for this type of operator learning work, you cannot extrapolate. So you're absolutely right. And this is the case for all these methods. I'm yet to see something that can generalize to everything. So the idea is this: you have a certain class of problems, you have training data for that class of problem, and then you learn only for that specific class of problem, how do you convert it to a simpler problem? That's it. You cannot, there's no free lunch. You cannot do this for extrapolate to any class of problems.
I see. You answered very nicely. Thank you so much.
Yep.
Any other questions? Well, so I'm sure that I imagine Amir also is available if anybody has question, if they want to directly reach out to him. But I guess if there aren't any more questions, let's thank both of our wonderful speakers. Thank you so much for sharing your knowledge and talking about the advancement in the field. Thank you so much, and I guess enjoy the rest of the evening wherever you are in the world. For Paola, evening, and then for us, morning. Thank you so much.